Hello.
Hi.
Hi. How are you?
I'm good. Over here, yeah. Were you able to find the paper reference I sent to you? It starts off with geometry.
Okay.
It's a good paper about development and epithelial tissue and how that interplays with development. Well, I've got reentrant rigidity transition in planar epithelial with volume and area elasticity.
All right. Yeah. So I'll go over that.
Yeah, it did. Well, it's got ordered and disordered and calmer and to flat tissues. And yeah, it just looked interesting.
Okay. So why don't we get started? So yeah, thank you, Susan, for that reference, and we'll look at that reference and we'll talk about some other things here. All right. So yeah, we'll do a couple things here. The first is this paper Susan sent out, and this is I'm just bringing up the arXiv stub here. Sarkar and Krajnc, reentrant rigidity transition in planar epithelia with volume and area elasticity. So this is the paper. Let's do the abstract and then we'll get into some of the figures. So we recover a rigidity transition in 3D epithelial monolayers described by cell volume area elasticity. So this looks kind of like the surfaces that we like to talk about, these epithelial monolayers, which if you look at sort of a cross section of it, remember they're these square, kind of like this, and they're kind of arrayed like this. And then 3D, we're just looking at this part here, where we have some depth to this layer. So this is the monolayer here, and then this is the depth, and then these are the interaction points. And we've talked about these trijunctions. So you might have here, this point back here might be a trijunction. This point here might be a trijunction if you had a cell in, this is no longer a monolayer, or, it has more cells in it. So this is like kind of the basic unit of what we're talking about here. It's a nice model to look at some of the properties of cell packing, cell populations. Looking at the monolayer, you get these nice mechanical properties arrayed along there. Okay, so we recovered the rigidity transition in 3D epithelial monolayers described by cell volume area elasticity, or VAE. So this is where we have our volume and our area. So these are just two measurements of each of these cubes, and we get this measure, which is VAE, and it has something to do with our volume and our area. And then we're measuring elasticity, which is this property of deformation, basically. So the deformation is when we put some pressure on this monolayer. How does it deform? How do these, where the cells meet, how do they interact? And so it has a lot to do with these trijunctions. And in this case, it would be a bijunction, would be kind of the normal mode of things. And then you might have a trijunction or an end junction that would also, exhibit some interesting properties. An in-plane isotropic strain drives a reentrant columnar to squamous rigidity transition. And so they talk about reentrance. And, this is, I guess, kind of this property. You hear this term a lot in neuroanatomy. I think it's basically feedback. But, they're talking about this sort of property again of this entire sheet, not just single cells. It's describing the sheet plus the interactions when a strain or a stress is applied. With a critical point near the unit cell aspect ratio. In addition to the vanishing shear modulus, the phase diagram also features floppy states with zero in-plane bulk modulus and a discontinuous columnar squamous transition controlled by the lateral tension. Our results provide a 3D context to the rigidity transition to the well-studied 2D area and perimeter elasticity, or AP model of epithelia. So this is, of course, we'll get into this, offering a resolution to the counterintuitive compression-induced tissue softening predicted by the 2D model. So we'll get into this figure in a minute. I want to mention a couple things from the introduction here. During morphogenesis, epithelial tissues must have an ability to transition dynamically between elastic and fluid-like states. So, there's this elastic state and this fluid-like state. So we have this sort of state transition or this phase transition that's queued up. But we know that soft tissues have these properties that are, allow them to not be rigid. So, purely mechanical systems are rigid. And the problem there is that, if you model these if you use rigid system simulators to model biological tissues, sometimes you can run into problems that they don't, your model doesn't behave like a tissue. What they're doing here is they're pointing out we have these elastic and fluid-like states, and we have to have this sort of ability to transition between these states. This allows the sheet to support mechanical stresses on the one hand, but also facilitates cellular rearrangements and large-scale tissue flows on the other. So we want to be able to, this cell layer, we want it to sort of contract so that they can be packed very tightly, so they can be very narrow. You can pack it in very tightly. You also might want to have other types of deformations like this, So the cell sheet is kind of impacted by these different forces. You might have something like this. You might have forces like this. Or, you might want to have them break out of this sheet and start to flow, or you want them to be fluid. And so that might mean where, one cell might migrate upward against the rest of the sheet. And you might have like a flow like this, where cells are kind of moving away from one another. And of course, you have a medium outside of the sheet of liquid, say, or some sort of flow. And the cells are embedded in that, so they're responding to those forces as well. And so, you have all these diverse behaviors. And so that's what we're kind of trying to account for. So there's this dual solid fluid behavior. People have studied this using the APE model, or the area and perimeter elasticity model, which assumes that each cell's apical area A and perimeter P tend towards preferred values AO and PO respectively. So if we go back to our cells again, we have this APE model or APE model. And what they're referring to here is if we have a cell, we have the apical aspect here, which is A0, and then we have the perimeter, which is P0. And we can do that for each cell. We can do that for the cell sheet. And we can get this elasticity. I guess that's a coefficient. And tells us something about how these cells are kind of transforming in terms of shape. So it's a shape description, and there are probably better ways to do it. There are a lot of ways to do shape descriptions, but this is, I think, a good approximation. Okay, so this model assumes that each cell's apical area and perimeter tend towards preferred values So A0 and P0 are the preferred values, sort of the stable states. The model predicts a rigidity transition controlled by the preferred cell shape. So that means that tissues are rigid when the actual cell shapes are incompatible with the preferred one, and floppy when cells can attain their preferred area perimeters. So basically, when it's outside of its stable state, the tissue becomes rigid. So you not only have one cell, you have a bunch of cells. And what we find is that when they're not in their stable state, they become rigid. So if they're in like this state, they might become rigid, lest they kind of come apart as a tissue. Or if they're in their preferred state, they might become flexible. So this might be its preferred state. Maybe it's a relaxed state, and then you get these kinds of this ability to be flexible. So that suggests that there are maybe a lot of things happening at the tissue level as well as at the cell level to keep this relationship stable, because you don't want your tissues to fly apart under conditions that are not preferred. But then when you're in a preferred position, you want to have that flexibility. And again, this is just a property of the mechanical aspects. So that's what we're dealing with. Okay. In the floppy regime, the linear shear modulus vanishes, and depending on their structure, tissues may also display zero energy barriers for cell rearrangements. So this is where we're in this floppy regime where we have our preferred areas and perimeters or preferred shape. In this regime, we have no linear shear modulus, and so we can rearrange our cells as this is not, we don't do that, but the biological system can rearrange things very freely. These findings extend to fully three-dimensional space, filling cell aggregates. Analogous to 2D, the generalized 3D model employs volume and area elasticity, or VAE, which minimizes the energy when cell volumes and surface areas match the preferred values V0 and S0. So this is similar to APE, which is the volume and area elasticity as opposed to area and perimeter elasticity. Okay, so then we have these two different models. We have the two-dimensional model, which is APE, and the three-dimensional model, which is VAE. So we have our APE, which is two-dimensional, and our VAE, which is three-dimensional. If I understand this. So this perimeter is a description, actually, of, I guess, this area here. So, okay. All right. So, as in 2D, the preferred 3D cell shape controls rigidity, and the linear shear modulus vanishes when tissue transitions from rigid to floppy states. Okay, so while 3D VAE models naturally extend the concept of rigidity transition to bulk cell aggregates, it remains less clear how 2D APE model translates into 3D epithelial monolayers. So how do we get this two-dimensional model transitioning into this 3D model? A recent model developed by some of us addressed this open question by coupling an apical cell mechanics described by APE to the basolateral cell region under surface tension. This coupling inherently introduces apical basal asymmetry in cell mechanics, suppressing the rigidity transition such that tissues remain rigid regardless of the preferred apical cell shape. So this is figure 1. This is a simulation snapshot. This just shows ordered and disordered phases. So Susan mentioned that they talk about ordered and disordered phases in the paper. This is this phase transition. So you get the ordered, which is here. So you see the way they've modeled this as a cell sheet that has this depth. So, we have this model here where we have the APE model where we have the perimeter of a cell, and then we just would have this kind of depth to the cell sheet here. So keep going out like this. And, we want to know kind of the constituents of the monolayer are back here. So that's what this comparison is. And we have the ordered phase, which is here, which is where they're tightly packed and they're kind of aligned very neatly. And then the disordered phase.
Sorry, they're hexagons.
They're hexagons.
Yeah.
And then the disordered phase, where they're kind of out of phase in terms of their alignment. They're hexagons, but they're disordered. So you see the packing is.
The disordered ones are probably more that some of them are 5-sided and some of them are 7-sided, because those two will do a tiling.
Okay.
And the 5-7 tiling ones are more fluid.
Oh, okay.
Yeah.
So the 5-7 tiling ones would be in the disordered phase, or in the ordered phase.
Yes, and of course there's other ones, like you can do 3-8 or something as well, just depending. And that goes, this looks like it goes for the basement membrane as well as the top apical part of the tissue. And sometimes for the any of these, the top tends to move around more than the bottom, and you'll see different structures on the top than you have on the bottom.
Okay. Yeah.
Well, you can see that the top can move around a lot.
Yeah.
Where they're attached to the bottom. So yeah, that changes as well.
So yeah, we have the basement membrane down here, which is, and then we have the tops, which are the apical end up here, and that's where you get this movement. So you get like deformations kind of along this, so you might get some bending here.
Yeah, and twisting too, because you get scatoids to twist too. Yeah. So yeah.
Okay. Yeah, so that's your ordered and disordered. This is rigid like columnar. This is floppy like cuboidal. So this is where we have the same kind of thing, where we have ordered and disordered. Then we have rigid like squamous, where we have the ordered and disordered as well. So they're doing these simulation snapshots of these sheets, and they're just looking at, so it says, for clarity, around 10% of all cells are shown. The fraction of non-hexagons in disordered sample, D equals 0.437. So I don't know if that means that they, in the disordered sample, they have, I guess, a parameter value for non-hexagons. So they have a different fraction of non-hexagons in the disordered phase that are here. So you can see that, like Susan said, they're not just hexagons in the disordered phase, but other types of shapes as well. Figure 2 are these two-dimensional simulation snapshots of this transition in ordered tissue for SO 5.75 and disordered tissues for SO 5.9. So this is where we can see some of these. We have this epsilon value, which goes from 0.2 to 1.0 to 1.9. We have the ordered at the top and the disordered at the bottom. So you can see for range this parameter sweep what those changes look like. You get this, I guess the ordered, it's sort of, it's a hexagonal lattice, a purely hexagonal lattice. At a higher value of epsilon, it's a little bit sort of, the tiling is aperiodic, and then you go back to a periodic tiling at 1.9. And then for the disordered, I guess you can also recover this. It's aperiodic, I guess, and then it's it's more aperiodic, and then it recovers that tiling. And then so this just shows the phase space here, where you have this floppy regime, this rigid regime. There's a critical point here. This is between, I guess, SO and epsilon for these different values. you get the ordered regime at the boundary between rigid and floppy, and then this disordered regime within floppy. Okay. And then this just shows some other aspects of the phase space. So I guess this is the important figure 2D, the important thing to point out, which is that there's this floppy phase, this rigid phase, and then this critical point, and then this boundary between order and disorder. Okay. Yeah. And then we go down to figure 3, we see total energy per cell. We actually see this difference here between squamous and columnar. So in 3B, and this is just an energy profile in A, but this shows this difference here, this sort of phase transition between squamous and columnar. So squamous is where it's pushed, it's compressed in terms of its height, and then columnar is long, has a longer z-axis, I guess. We get this value of epsilon where it's stable, and then there's these degenerate states at the phase transition, and then you get for positive values of gamma, I believe this is the columnar state. So we reach a 0 value for gamma. We go from negative gamma to positive gamma. There are these degenerate states right around this phase transition, and this is a transition from squamous to columnar. And so that's what they're getting at there.
I'm sorry, the stress strain curves for squamous were different from the columnar
Okay.
in my measurements.
Oh, really?
Yeah, they behave differently.
Huh. Okay.
As a sheet. Yeah.
So
this is my model.
Oh, in your model. And then what's the difference between what they're doing and what you're doing?
You realize I have over 200 graphs, right?
I know, yeah.
So, what's the difference? It's how they, let's see, how they behave in compression as opposed to tension.
Okay.
I don't know, squamous cells are very interesting.
Yeah.
Yeah, I could look it up. I could once I get this paper written on it, I'll present that.
Okay, that'd be good. Yeah, that'd be good. Okay, so yeah, then they have their discussion. Our results show that the rigidity of epithelia with VAE may be tuned non-monotonically by in-plane isotropic strain, describing a reentrant CST rigidity transition, or by tissue transition from rigid columnar to rigid squamous states. Their intermediate floppy cuboidal states exists. And additional constant lateral tension, distinguishing lateral from apical basal sides, introduces a discontinuous columnar squamous transition, which we saw in figure 2, or actually figure 3, with energy degenerate states with 0 in-plane bulk modulus, where lateral tension is absent. So our 2D project model shows that varying 3D cell aspect ratio, which is by imposing in-plane isotropic strain, effectively corresponds to nonlinearly changing the effective in-plane preferred cell shape index, P0, which in turn controls the rigidity transition. This provides an alternative physical interpretation of P0, which has been so far interpreted through a competition between cortical tension and adhesion strength. And then the 2D8 model, which inherently does not describe cell height, counterintuitively and in contrast with the standard jamming, predicts tissue softening upon in-plane compression. By accounting for a third dimension, our model changes this view, indeed compressing and dilating a monolayer away from a cuboidal state. Both leads to its rigidification. So this is where they kind of differ from some of the things in the past. They use this third dimension and they get the same interpretation. Our analysis shows that the VA monolayer model is a more direct 3D analog to the 2D ape model than the model in which apical cell sides describe an ape are coupled to the basolateral cell domains under surface tension. Indeed, while the apical basolateral coupling in the latter model breaks the apical basal symmetry, preventing the rigidity transition altogether, our 3D VAE monolayer model preserves this transition and in the limit of incompressible cells, even exactly maps to 2D fixed area ape model. So, yeah, they mention here, while the assumption that epithelial cells tend to preserve both their volumes and total surface areas, that this is yet to be experimentally validated. So the models really kind of make this assumption. This phenomenology may readily apply to studying synthetic planar packings of strongly adhering lipid vesicles, where the membrane's bending elasticity may be negligible and the mechanics may be well described by the VAE model. So that is that paper. Thank you, Susan, for pulling that up for us. Yeah. So I want to mention one more thing, and then I'll get to Lalith. So Dick put out a new paper in BioSystems. This is brand new. So Dick, of course, Dick Gordon is a member of our group. And I think we've talked about this in the meeting, but this is something that he has, I guess, been asking. Is there a relationship between embryonic bioelectric currents and differentiation waves? So if you're familiar with sort of this idea of bioelectricity and development, this is something that has a deep history in developmental biology, and most recently Mike Levin has picked up the torch on that, and they have now, of course, a journal devoted to bioelectricity. And the idea is that bioelectricity plays a role in morphogenesis. So you have these, cells can generate bioelectric currents. They can generate bioelectric signals. Of course, we know about action potentials, and those happen in neurons. But you have all sorts of cells producing different bioelectric signals. All cells have ion channels, and they produce some sort of measurable trace. And in the case of action potentials, they have a spindle, and they have all these properties that are unique to action potentials, but you actually get other types of signals as well. And so, then the question is, what are they actually doing in development? And so the answer, I guess, is that they're coordinating cells. They're creating fields that can be, beyond the single cell, and they can set up different morphogenetic events. They also may play a role in wound healing and other types of regenerative capacities in tissues. And so what Dick is asking the question of is, are these embryonic bioelectric currents that will surely result from these kinds of interactions? Do they have play a role in setting up these differentiation waves? So the kinds of differentiation waves that we've talked about in a number of papers that Dick has been talking about for years, do they actually have a relationship that's interesting? And so I noticed that Dick has been publishing a bunch of these kind of theory-related papers in the last year or two, where he's asking questions and posing them, and it's not like we have a good answer for them, but it's that he wants to get those questions out there, and people may be working on them. But he's posing the question. So the abstract is short. It reads, differentiation waves usually spread in embryonic epithelia until they reach boundaries. The stopping power is but unexplained. So differentiation waves, in short, are these waves that are propagated through tissues or these sheets of cells. And so they may originate in a single cell, and they send this wave across cells, and the cells are getting this, basically the stimulus, to differentiate. And there have been different mechanisms that Dick and colleagues have explored. There are all sorts of mechanisms. There are molecular mechanisms. There are mechanical mechanisms that affect cell division. All these different types of things that are proposed. So we don't really know what exactly sets up we know what sets up a differentiation wave. It could be a chemical signal, it could be an electrical signal, it could be a mechanical signal. But then when an individual cell gets that signal, what's the mechanism by which it's transduced into the cell itself, and then it's able to produce this result in the phenotype, which is that the cell differentiates or otherwise changes its morphology or changes its relationship to a tissue. So we did a nice analysis of this in Drosophila imaginal disc, looking at the imaginal disc as there's this wave that goes through this furrow that moves through the imaginal disc, and it changes this imaginal disc from this disordered tiling, which we saw in the last paper, to this set of ommatidia, which are going to form then the eye structure. So there, we can have these waves that go through tissues. They can enable this kind of differentiation. We want to know if bioelectricity has a role in this, and if so, what is it? So differentiation waves usually spread in embryonic epithelia until they reach boundaries whose stopping power has been unexplained. Bioelectricity in embryos has two ranges, cell to cell and long range. So you have, like in bioelectric studies, usually you have sort of these local and global effects. So we might have a sheet of cells. Again, we might have like this set of, we'll just do some hexagons for the sake of making this simple. And we have the sheet of cells, and we might have a signal that originates in this cell down here, and it sends its signal outward, and these are the cells it's going to affect. So you can have the cell-to-cell relationship. So cell A and cell B might have a relationship. There might be a reciprocal relationship where you have something that A is affecting B, B is affecting A, and there's this coupling. And that's a local relationship. And so that would be A to B coupling. And then you might have a more regional or global relationship. So in this case, you'd have A, B, and then C and D. And you'd have this relationship here where the signal propagates out. There's this signal that propagates maybe linearly with decay. So the farther you get out, the less signal you have. And so it affects maybe this global or regional signal. It affects a host of other cells. So A to B. And then you might also have C and D, which are a little farther away, and they might also be affected, but not as strongly as B. But you get the picture that there are these different levels of effects. And so we want to understand maybe, this is how you get, different effects at different length scales. And so there are a lot of physics implications for this. Susan?
Yeah, it's a cell cramp. I actually have a picture of one on the surface of an axolotl.
Oh, yeah.
And it definitely is electrical and mechanical, for sure.
Yeah.
Probably chemicals. It's calcium. You cause a epithelial cell to cramp with calcium because it works like a muscle. You've got actin and myosin, and if you add calcium, then it does its ratcheting motion and causes the area to cramp, right, to contract. So you have these cramps coordinate eventually and become a wave of cramp that go up the side of the axolotl salamander egg and in some of these insects that you can see. And you get what Dick is calling a differentiation wave, because after a cell has undergone the cramp, it's rearranged its cytoskeleton into, say, a more columnar configuration, and maybe it relaxes back down, but it's rearranged its cellular components, and these waves last long enough that it rearranges the nucleus. It changes the shape of the nucleus, and that changes the gene expression. So that's all the theory there. Yeah. But I have pictures of a cramp.
Oh, wow. Okay. Yeah, that's good. Yeah, there. We have pictures available. Dick has some in his books, and I know you've probably been playing around with it. And we did a review of this in the BioSystems paper we published last year. So there's basically we're interpreting a lot of not just signals from cells, but you get like calcium waves in the embryo, and they also can be kind of interpreted as differentiation waves. And so you can observe these empirically. We can understand that this is a theoretical concept where we have many different types of activity, and then we have this is it's a way to kind of think about how morphogenesis proceeds, not just in one cell, but in a tissue where a bunch of cells are linked together. Yeah, and then go ahead.
The thing is they are slow, so people haven't noticed them because it's you do a time lapse or you slow down the movie. They occur over hours, so you can't really personally you can't see this. But if you take images and then do a movie of them, you'll see that it really is a moving wave. Actually, I was going to take pictures with one of the microscopes that I have just of the top of the egg so people can see this. Like Dick lost his movies. It was a miscommunication or something. But I have a microscope that would just simply, if I just put an egg under it, that it would take pictures with a proper 24 megapixel camera and get really decent resolution of it. So I'm just going to take a picture of the top of it, and my flipping microscope decided not to work. So rather than fix it or anything, since I'm busy, I'm just going to take pictures of the top of the egg, and I should be able to see some of these waves.
Okay, that sounds good. Yeah. Yeah. So there's this temporal aspect, there's a spatial aspect, and so what he's trying to do here is get a sense of how this problem can be solved, basically. So it is postulated here that the long-range component somehow alters gap junctions in intermediate filaments, creating the boundaries for the trajectory of differentiation. So if in these cells we have gap junctions, we have intermediate filaments, which are all things at the boundary of two cells, where sometimes you could have things that are propagated from A to B to C to D, and through different means. So there are all sorts of ways in which these waves can be propagated. A combined EIT electric impedance tomography / visual microscope is proposed to investigate this proposed correlation. So he lays out kind of what needs to happen for this. And he talks about sort of an example of mechanical initiation of differentiation waves. This is one that is observed to correlate with primary neural induction. The blastocele is known to come under pressure. We have shown how this might launch differentiation waves during primary neural induction. The artificial release of pressure in xenopus leads to multiple neural plates. And then it is possible to organize differentiation waves as differentiation trees, subsuming lineage trees, whose evolution has been postulated to explain heterochrony, macroevolution, and metamorphosis. A differentiation code has been postulated to be the genomic basis for these trees. Heterochrony can be explained by variation between species and the timing of branch points in the differentiation tree. So this is where, heterochrony is sort of the timing of growth and the timing of differentiation. And so if we go back to our differentiation tree, which is derived from the lineage tree and actually has more information than the lineage tree. And I'm not going to get into drawing out a differentiation tree now, but you basically have this tree, this binary tree, and the length of the branches correspond to sort of the length of timing of this process. So this wave might be fast or slow, and then it leads to this sort of change that can be, described using this heterochrony concept, where you get changes in the swiftness and degree of differentiation. Macroevolution may correspond to topological changes in the differentiation tree by a copy and paste of portions of the genome. Metamorphosis may be the result of rare concatenation of two differentiation trees via matings of different species. And he's talked about this problem with, where, Williamson has this controversial proposal that different genomes have mixed over evolution. So you get like different parts of genomes that kind of lead to differences, that sort of the DNA of different species have kind of intermingled through transposon means or horizontal gene transfer or something like that. And so you have this ability to take different, basically differentiation tree programs and concatenate them into the same differentiation tree. There are other, mechanisms where you're going to have the evolution of these different programs that lead to differentiation trees and so forth. And so then he gets into bioelectricity, and then he talks about combining the two approaches to embryonic development that involve bioelectricity and looking at differentiation trees by suggesting that the long-range bioelectric currents alter gap junctions in a way that halts the propagation of differentiation waves from one tissue to another and thus limits their trajectories. So using these kind of local connections, gap junctions, things at the membrane, and then getting this propagation from cell to cell instead of like some sort of global signal where, it might be like a mechanical or a chemical signal that kind of gets propagated out, broadcast in this way, that actually has an effect on limiting the trajectories. And so this is I guess a finer grained approach to this idea, and so it requires specific types of measurement, and so that's what he's proposing. So he gives a number of examples here in embryos. He kind of goes through the background alternative models, and so this is just kind of laying out the problem space and how you might approach this. So that's a nice little sort of hypothesis theory paper. Congratulations to Dick on that. All right, so okay, so we have some references by Susan. This is the one we talked about: reentrant rigidity transition in planar epithelia with volume and area elasticity. This other paper by Lang, Matos, and Key. Vertex models capturing subcellular scales in epithelial tissues. This is a PLOS paper. And then Mehul said it had to leave, got an insect bite. So hopefully Mehul's okay. Okay, now Lalith, you wanted to present that last part of the presentation.
Hello.
Yeah.
Hi. That was a great work by Dick and like very insightful presentations from the previous ones. How much time do we have, Bradley?
We have as much time as you want. I mean, maybe, like not an hour, but.
Sure. I'll quickly try to discuss. So I made little changes in the presentation so that I added little more diagrams.
Okay.
I thought the slides were a little emptier last time. So hi, I am Lalith Baru from IIIT Hyderabad, Brain Cognition and Computation Lab specifically. And yeah, so I'm gonna work on this for the next couple of months under the INCF, like Orthogonal Research Lab and Education, under the guidance of Professor Bradley and Jesse, Mehul, and others. So yeah, so to give a fine introduction, like background and the motivation. So the motivation is like the C. elegans is one of the smallest organism nematode worm which has like very limited number of connections, limited number of neurons, and so if we are able to develop like neural networks on these small networks, like small possible modeling, if I can do this, then we can see a bigger picture, like more biologically plausible AI, and we can design more enhanced and deeper neural networks. So that's one of my inspiration to delve into this. And the neural developmental is a very high dynamical and temporally extended process where it's not just extends in a simple division manner, but it has a certain cell division pattern and it evolves in a lineage tree in a very hierarchical fashion, and the spatial locations and the migrations happen in a very complex fashion. When we try to model this, these hierarchical interactions and multicellular interactions that actually evolve over the time, then possibly then we believe that we are taking a one step ahead to understand the how the organism is developing from a single cellular to like a multicellular organism. So that will give us a very much insights into like how the tissues have been divided, how the neurons have been segregated into like certain the sensory neurons, the motor neurons, and how are they divided. So we can understand more into details about all these things. And in the next, so when what we believe is because it's a higher order interactions, and multicellular organisms depend on hierarchical functions. Like when we try to model these using hypergraphs, so when we try to model these using hypergraphs, we believe that these higher order interactions can be captured in a very good manner rather than a diad or a static graph. So this was the main motivation and the basic introduction of what is it. The project is divided into 3 preliminary objectives. First, we design the foundation, that is, we design how we want the the structure of the the data that we want. So we lay the foundations in a way that we structurize data in certain format that so whenever we use a different hypergraph networks or we use different neural network, or when we use a different we can use this framework in such a way that it can be a nice plug and play. So when we keep the foundation right with the data, then it could be one of the good. Second, we are interested in the unification, because when we have the data, then the fundamental idea is to this can be applied to a different models. Suppose we try to understand the development of a certain C. elegans. So, but it should be replicable for various other nematodes which have been taken birth. So not just one time single organism data, but it should be useful for multiple. And whatever the model that we are trying to use, I mean, like we already knew that, so the kind of data that we get from C. elegans is more of an invariant, but still we need a unification framework where we need not additionally add certain layers or certain neurons, but it is holistic in itself. And the third is we want to see how this emergence happens when we use this framework, as when we vary the layers, when we vary the the message passing mechanisms, when we vary this I mean, like the number of the neurons that we use, or the type of the hyperedge connections that we use. So like, how is the emergence happening? So this entire framework helps us to understand this. So we divide, I mean, like I divided it into 3 phases based on my understanding and the way I can construct this project in a more better manner. So the data unification and the dynamic hypergraph construction is the first foundational step. Second is the unification of hypergraph, the message passing, the architecture, the layers and the weights, and they all can be unified into a single framework where we can just plug and play for the performance and the emergence, where we see that how the connectomes and the functional modeling happens. So it's basically the outcome. So yeah, so and the previous we saw that the taxonomy in one of our previous videos, where we see that modeling of higher order interactions can be taken in, like how we are taking the input features, how are we encoding it in an input structure, and later we saw that we can use different message passing mechanisms and certain way of learning these messages. So this is the whole taxonomy of how we're going to do. So I divided into like 3 dimensions, where the first is how do we construct an hyperedge and a hypergraph, where we include all the lineage, spatial, function, and all the different other features, and also use a different message passing mechanisms, where we not just use a simple local or a global attention, or like summing up the features, or having a mean of all the relevant features, or can we apply certain attention to the features which are more relevant, or these type of message passing mechanisms we can include it in. And the temporal dynamics. So the previous works morely they focus more into the static frameworks, which cannot be used in real time. Whereas in growing topologies, there can be a cell division which strongly that can happen, and that can lead to the which can be fatal. It can lead to a cell fatality. So these things should be captured while the modeling. So the static nature of the graph could actually impede this thing, but whereas this hypergraphs allow us to model these dynamic topologies and the growing topologies. And so I wanted to give a short quick and anyone has any doubt, you can just stop me.
Okay.
So the hypergraphs and hypergraph, just to give a basic introduction. So hypergraph is basically a generalization of regular graph, where the edges are certain hyperedges, where they do not restrict to a dyadic connection, that is a one-on-one connection, but it can connect to any number of nodes, not just 2. So here we see a simple graph where V is vertex or edge, V is vertex or the node, or E is edge, where we represent in this as V small I are basically number of nodes which in a given whole subset of capital V, and E small E subscript I says that it's edges. And whereas coming to hypergraph, we denote it as H V. The number of nodes are same, but the edges are different. I mean, like the connectivity is different. Where epsilon I is basically the hyperedge we can see, and this hyperedge can carry any information. So I have here given an example where XYZ are the coordinates of our C. elegans and phi is the phase angle. So just to give an example, these all information can be fed into the hyperedge so that the modeling could happen. And so when we try to understand the mathematical intuition, we see the hyper node actually connects multiple nodes instead of a single node. So that's a hyperedge. So here red color box, red color oval is basically the A, the B, the C are the hyper edges which are connected to all these nodes. And one can expand it in like two different fashions. One is clique expansion, so which might not be useful because we cannot retrieve this clique expansion back into the graph. But whereas the star expansion we see in the right the third one, which is it more of like an entity, and so node is considered as the features, I mean like the connections, and ABC are basically entities. Here the entity ABC are the kind of hyper edge connections that the nodes consists of. So it's basically the star expansion. That's one of the way that we can model the hypergraph. When we convert a hypergraph into star expansion, then it is possible to convert the star expansion back into a hypergraph. So that's one advantage. So yeah, so so we are trying to understand a simple mathematical intuition where here, as we discussed earlier, so V is basically the number of nodes that are present, and small e is the edge. And what this basic function here tells is, here the h belongs to the the layer of a hypergraph, where it multiplies with a weight vector and then it adds. So basically this is a kind of and each summation happening at each and every time means that it's actually aggregating the information by adding a certain weight to that specific set of the hyper nodes. So here this expansion, this is basically one of I mean like the intuitive example of how a hypergraph can be captured. And here t+1 is a time step. So each and every time step while it's evolving, so it captures how is it growing, and the growth can be captured using this basic network. So this is the mathematical intuition. So this is a step one, or like it's kind of a hello world to kind of hypergraphs. So yeah, so here when we represent this as like this is a dynamic hypergraph representation. So this actually models complete developmental process where each sequence it carries a node starting, or a node which has like a bunch of features, and it encodes all these lineage depth, the spatial coordinates and molecular state and everything into these features, and then it's carry forwards into the time. So here time t and t+1 reminds us to like each and every time step how the graph evolves. So that's why you call it dynamic. And the interactions that the functionality that it has, that the summations and the I mean like the non-linearity that is applied on these functions basically tries to capture the developmental events here, such as the way the cell is divided and the type of centrifugal fusions it's happening, and then the spatial neighborhood relationship that's actually acquired during the period of time. So these can be captured using these hypergraphs. And so what I mean like I proposed during the project is, so how can we model this growing hypergraph neural networks? So as we have seen, in the time it has grown, I mean like it moved from time to time the way the hypergraph evolves. So we call it as like growing hypergraph neural networks. So the main advantage is the dynamic addition of the nodes and the hyper edges, because the hypergraphs have an advantage to learn these nodes very dynamically at every step. And the second is we are trying to integrate all the information that has like lineage, I mean like spatial, the functional cues, and also like the phase angle relationships and various other things. And this is more of a learnable, and this is a data driven simulation. That means if we have a data of like 6 to 7 C elegans developmental process, then maybe we can simulate a more, I mean like if we have like a 10 to 20 examples, we can make the model learn that okay, this is how it starts from a single cellular to a multicellular organism, and this is how the spread of each and every the cell divides, and it this is how the lineage tree grows, and this is how the spatial coordinates move in order to finalize into a organism. I mean like and that's how the developmental process can be captured. With like 20 examples, we can simulate like 100 more. So by learning them. And yeah, just as an example here, the AB cell divides into AB, A and ABP, and the lineage tree again grows in the second time point division, where ABA divides into ABAL and ABAR, and then ABP divides into ABPLN and ABPR, and this goes on. So as we have seen that, initially the coordinates of AB have been at a certain location, but they have been dispersed during the developmental process. And this also can be captured. As we can see that this is just a two-dimensional example, but we can move deeper into things by three-dimensional visualizations. And even in three-dimensional visualization, we can see that there are change and the shift in the coordinates. And this is the movement happening. The way the organism has to, it swirls around itself and finally becomes a worm with a head at a certain location and a tail at a location. So all the events can be captured by this network by modeling all these time points together. So this is just an intuitive example saying that okay, the cell has been divided, and the red indicates the first division, the green indicates the second, and the blue indicates the third division. So this is just an intuitive example, but this can be applied to a graph neural network where it can understand the complete evolution process. So yeah, so when I include the phase angle part here, we can see that first the cell was divided with 198.9 degrees, but later the divisions were like smaller in number. So we can see the change. Why it was divided into 198 and how it divided into that. So we can get the three-dimensional coordinate exactly, and we can get the the movement where it starts from and where it ends from. The spatial locations can be captured more accurately in these scenarios. So how did I design this timeline is in the first 2 weeks, I'll see how we can set up the things, the data sets and the literature, what we're going to work on, and the complete design if it's ready. And if it's, agreed with the mentors, then we can move into the next step where we can, design the hypergraph and integrate and see how it can, work with, dynamic growth. And later we can add these message passing mechanisms, and we can evaluate how these message passing mechanisms are like helping the model to learn. Not every message passing mechanism might help, but we have to understand which one is actually good for it. And in the 7 to 8, in the end of the second month, we'll probably see that how this temporal modeling can be scaled. Suppose I have a developmental data of like 1 C elegans. Am I able to understand the 2, 3, 4? The other set of developmental process of like other C elegans. And later we can see whether the connectome that is being developed and the fully trained network, if the network is well trained, and when I see that network, when I'm trying to understand that okay, this when I start from AB here, how it can evolve, the one way it can evolve, and is this connectome aligned with the what is the actual thing, so that we can observe and we can have the nice visualizations and check okay, the modeling one has been performed like this, and the original is like this, so we can see the connectome alignment and visualizations too. And in the final step, we can do the, if we are significant enough from the existing literature, and if we are able to make a big difference, then we can document it in a very good manner. And one, we can do a final submission to the the GSOC, and also we can write a good piece of work where the community actually see that there is a good improvement in this space. So that's the main motto of doing this project. So the contribution, how could I contribute to INCF is first designing a unified model. So this could be very much helpful because existing works do not capture all the relationships into a single framework. And the second is when we use this hypergraphs as a developmental when we apply this hypergraph into a neural developmental program, then this adds a very high weightage to understand that, in a further piece of works, because the existing works focus on just like designing the connections and just visualizing them. But this can be leveraged to learn the complete patterns of like starting from a single cellular, organism to a fully multicellular organism. And the pipeline could be reusable for the further research, and visualization tools can be used to not just to visualize the hypergraphs, but also to visualize various other things, like how the like an example I have shown before, that how is the variation that is observed when the cell division happens from ABA to ABP. So yeah, so in future work, what I believe is, how can we extend is, so once we understand the certain mechanisms of like how the entire pipeline works and all, so maybe we can take some design principles and take some motivation from here to design a new line of architectures in the space of AI, because AI is more of scaling these days, but there is no underneath biological principles like they used to be previously, because convolution neural network has a little bit of biological principles, but the later set of models are being used to feed the data. But if we can understand that, okay, feeding the data can make the network learn, but why? So these design principles can be extracted. So by understanding the living creatures, starting from, let's see, elegance. And we can design biologically plausible neural networks, because once we start from like C. elegans, we can try see extending this to like a bigger brain, which is compared to those C. elegans is like Drosophila. And if it works in Drosophila, then we can extend, or we can see the limitations and build a better model where it can scale to starting from like like C. elegans to that of like human brain. Human brain is much more complex, but yes, I believe that one day we can do that. And one of the crucial thing I wanted to pinpoint here is the syntaxial relationships can also be useful, like the way the cell divides, that actually makes the syntaxium to form at that certain place. I mean, like when like in the previous discussion we were having about like epithelial cells and epidermal cells and all. So if there is like an epidermal synthesion that is formed there, so is it formed during the developmental process for like all the the C. elegans, and is there any connection that it forms in a certain fashion? We can observe these emergences during the developmental process. I believe that could be one of the good extension to understand those details. So yeah, so that's the end of the presentation, and these are some of the references. And yeah, thank you.
Well, thank you, Lalith. That was good. Yeah, we're looking forward to working over the summer on this and some of these open questions. So, I mean, you have your timeline. So we're starting next week on working on the coding period. We've been doing things with the community period. So now is the time sort of when we start doing this. And I know that I got in touch with Jayadratha, and he's also getting ready for everything. So
yes, sir, very much excited.
All right, thank you. Looks like Ibrahim is here from NeuroTechX, or would you like to introduce yourself? Okay, go ahead.
Hi.
Hi. Yeah.
Okay, cool. No, just dropping in. I'm working with Morgan in San Francisco. Recently joined NeuroTechX as a contributor, so I'm just a fly on the wall. I have nothing of value to say at this time.
That's okay. Welcome.
Thanks.
All right. Well, I think that's it for today. Did we have anything else? I know Morgan and Jesse joined late. Did you have anything you wanted to mention? Or
Oh no, no.
It's,
Go ahead.
go ahead, Morgan.
No, I'd forgotten about the meeting because of the holiday. So just juggling kids with this, so no problem.
Yeah, things I'm gonna say more next time or later this week, but the internship stuff's going really well. For other projects, I still think a lot about sort of the simulation agent computational stuff, and even some actual developmental bio-ish tie-ins of that, some of the time for that. And then applied to some interesting stuff at some other projects, which I may share next week that have a little bit of tie-in to some of this as well. But mostly good stuff, and good meeting, and excited for Google Summer Code. So thanks everybody for that. More next time.
Yeah, thank you. That sounds good. One more comment I wanted to make on some of the things that Lalith presented. He talked about simulating development and generating like hundreds of simulations using the model that he's wanting to work on. And we've tried to been, that's been one aim of kind of what we want to do in the group. We've done this in different ways. We've done like cellular automata. We've done other types of lineage tree modeling, and we even done this, I think, a few years ago in DevoWorm. In one of the projects, we used, trying to remember what we used, maybe like a GAN model, generative adversarial network, to model or to simulate images of an embryo. So you're working from a microscopy image, you segment out the features, and then you simulate that. And it was interesting, but again, it's not as rigorous as we'd kind of like it to be. So we're kind of have all these pieces in place, and there's a lot of value in these kind of simulations. When you get a lot of simulations of a single process, because we know that development has these properties of phenotypic buffering, these properties where you get a very stable phenotype given this process that unfolds again and again, this generative process. We know that to get viable phenotypes, you need to have that kind of stability, and we even have like epigenetic landscapes that describe this process as this process of canalization and buffering. And so, without simulating the genotype, we can simulate the phenotype in these different ways to see what that process might look like, to see what happens if you change certain things, and see what the viable phenotypes are. Because that's a lot of development is about producing viable phenotypes. So you get variation in developmental phenotypes, but generally those are, a lot of times those phenotypes are not viable. So they don't really count in the sense of like what the end process of development is. So just some parting thoughts on simulation and the simulation of development, why that's important. Particularly important, I think, for advancing some of the things we want to do, in addition to the tie-ins to OpenWorm and the simulations that they're doing of different parts of the worm, so like the biophysics, the connectome, and so forth. Okay, so that's all for today. Thank you for attending. Oh, go ahead.
Okay, yeah, I put the paper reference that I've been talking about in the chat.
Okay, sounds good. Yeah, we saw that, I think.
Okay.
All right, thanks for attending.